in the scope. And this is the latest bird research from WVU graduate students. So this year there'll be 3 West Virginia University graduate students, Andrew MacKenzie, who's a master's student, Spencer Parker, who's also a master's student, and Hannah Clipp, who is a PhD candidate. Together, their bird research spans multiple habitats from grassy wildlife openings to wetlands to mature forests. And there are several different focal bird species, including game birds and a diversity of early succession and forest birds. So each graduate student will give a presentation, and then at the end of that presentation, we'll ask that student questions. And it's always great to hear about the new research that's going on. So I'm going to turn it over to Hannah, and she'll get it started. Okay.
Yeah, so if you don't mind stopping sharing your screen, we'll get started with Andrew.
Okay. Oh, share screen. Sorry, Hannah. One. There we go.
Yeah, okay. That looks like it's working.
Yep, okay.
All right, Andrew, all you.
Can you hear me okay, Hannah?
Yep.
All right, sounds good. Hi, everybody. How's it going? My name is Andrew MacKenzie. Today, I'm going to talk to you about the wetland mitigation impacts on avian biodiversity. It's going to be an update from what I presented last year, discussing a little bit more about what we know regarding the biodiversity of my study area. I want to mention my co-authors, Darian Becker, Chris Latuma, Caroline Arantes, and Jim Anderson. So first, just a little bit about myself. My name is Andrew MacKenzie. Like I said, I graduated here in May of 2021 with an undergrad in wildlife and fisheries resources with minors in conservation ecology and agriculture and natural resources law. Dr. Anderson offered me an opportunity to stay on here as a master's student, so I'm currently working on my thesis. With my thesis, there's 3 chapters of proposed research, but today I'm going to talk to you about pre-active and post-assessments of wetland restoration on biodiversity. So a couple of these pictures are just some of the other animals I work with. There's an eastern box turtle that I found leaving my study site, and then a meadow jumping mouse that I captured during my Sherman trapping. I'm going to give you a background of the site just to refresh everybody, the restoration efforts that we have completed. Then I'm going to talk about our pre-restoration abundance and diversity, our active restoration abundance diversity, and then our post-restoration abundance and diversity. I'll wrap it up with some of the early indications and future research, and then I'll open it up for questions. So a background on my study site is in 2017, there was a renovation to increase the water capacity for the Reedsville and Arthurdale area to provide more drinking water to the community. This caused a pretty severe loss in wetlands. So this study site, Ruby Run, and then 2 other study sites were proposed to be restored as part of the mitigation project. Darian Becker collected 4 years of pre-restoration research. I was her kind of a technician at the time, but I went out with her a lot. And then I took over data collection in May of 2021. The restoration efforts that I'm going to talk to you about today occurred from June 2021 through May 2022. And during that time, we collected data for the active restoration period. And then I will talk to you about the post-restoration data that's currently being collected from June 2022 all the way through September 2020, September 2024. So this is just a brief visual abstract of what was done. On the left-hand side, you'll see there's a degraded wetland with cattle inside the area. You can see that the water's a lot dirtier. There's multi-floor rows all over the site. The second panel shows you the restoration efforts that we're about to get into. The third panel is roughly what the site looks like now. That's going to be our post-restoration time period. And then this last panel on the right-hand side is ideally what the wetland will look like in 30 to 40 years. So I'll start by talking about the bank grading and the herbaceous vegetation removal. As you can see, there was a significant amount of earth moved. This herbaceous vegetation that was removed was specifically targeted for invasive species, but of course, when you're grading the bank, a lot of other things go with it. Because it was previous pasture, it was mostly pasture grass, barnyard grass, things we didn't want in our wetland anyways. So while we were doing that, we added some erosion control. You can see in the left-hand side, there's some big previously downed logs that we're using to prevent too much erosion. On the right-hand side, you can see that there's some rock veins and a little bit of what resembles riprap. And then this also adds to the aquatic habitat, which is a different chapter that I'm looking at for my thesis. We installed quarry mesh, which is something that helps keep the seeds in the bank. It also prevents species that you don't want growing from growing up underneath. And then we seeded with native vegetation. On the right-hand side, you can see some black-eyed Susans, some bone set, and those big white pillars are the tree tubes that we planted. We also had a little bit of a fence extension. So if you look at the top right-hand side or the northeast corner, this little area, the fence was added onto a little bit to encompass more of the surrounding wetland. And then finally, we had our woody vegetation installation, and we installed roughly 2,800 trees. And so now that I talked to you guys about all of the restoration efforts, I'll get to the stuff you guys are most excited for, which is the birds. Our avian point count surveys are spaced apart at about 55 yards or 50 meters. In addition to them being spaced out roughly 50 meters, our point count survey radius is going to be 50 meters. Either once or twice a month, we will conduct 5 10-minute avian point count surveys. When we collect 2 point counts a month, it's between April and June, which we deem to be the breeding season, and the spring breeding migration season. And then these point count surveys began in February 2017. So last year I showed you our abundance data. I just want to refresh everybody's memory that the pre-restoration and active restoration, there was no change, or the bird abundance did not change during the wetland restoration. However, we did see an increased bird diversity during wetland restoration. So on the left-hand side is our pre-restoration, our right-hand side is our active restoration, and you can see there was a much higher diversity. And then just to kind of keep everybody up to date, our pre-restoration birds that were most abundant are listed right there. I have a panel of the pre-restoration on the left-hand side to give you an idea of what it looks like. And then I featured this song sparrow here because song sparrows were the most abundant bird that we had. For active restoration, our abundance of certain birds did decrease. So we saw a decrease in some of our specific species being as frequent, but we saw an increase in diversity. And when I was looking at it, it's because we had a qualitative increase in birds of prey compared to our pre-restoration. And then on the right-hand side is a song sparrow nest that was observed during the active restoration period. For our post-restoration, this is what I'm most excited to talk to you guys about today. So our top birds, abundance-wise, are still our song sparrows, our red-winged blackbirds, Canada geese, barn swallows, and American goldfinch. However, two birds that are showing up more frequently are our tree swallows and common ravens. So we are only about a year out of active restoration or a year into the post-restoration, but we're seeing a lot more tree swallows, a lot more ravens, and then I've also seen a lot more crows. For the diversity, I've seen a qualitative increase in the wetland and shorebirds compared to the pre and active restoration. So I've seen a couple of yellow legs, I've seen a couple of snipe, and then I've seen a couple of woodcock, and that's a lot more than I had been seeing in previous years. And then I featured the tree swallow here. And just to summarize all this, for abundance, there's not a difference between our three treatments currently. We're still collecting post-restoration research, so that may change. Our diversity, our active restoration had an increase in diversity compared to the pre-restoration. Currently, there are no differences between the other treatments. And then we did have a seasonal variation, which is important to document. So there's an increase in both abundance and diversity during the months of April through September. And that's important to know because the April through September time period is when we have our birds coming in and out for the migration. For future research, I'm going to continue conducting the abundance and diversity counts through September 2024. For the post-restoration, I'm going to continue my data analysis as I get more data every month. And then I'd like to start analyzing some more of our species-specific abundance and diversity levels across our three treatments. My acknowledgements are, of course, Darian, who kind of laid the groundwork for this, and then Dr. Latuma, Dr. Rantes, and my advisor, Dr. Anderson. And then with that, I will take any questions as you guys can look at this beautiful figure again.
So if anyone has any questions, you can throw them in the chat. So it looks like there's one from Jane. So how wide is the restoration area?
Yeah, so the stream itself that we restored is about 1.5 kilometers. We restored half of that area. And then the width of it varies from the stream to the fence that we installed is about 70 to 110 meters at the widest points. And then in the narrowest points, it can be about 20 to 25 meters from the bank to the fence. I see Donna asked if we planted buttonbush. We did plant buttonbush. The species of woody vegetation that we planted were eastern ninebark, eastern cottonwood, eastern redbud, our common buttonbush, our swamp white oak, our pin oak, and our river birch. And then the seed mixture that we used was just a good seed mixture for the ecoregion that had the consistency of the riparian wetland vegetation that we'd expect to see in our area. For Jane, are the ravens using the area? I have seen the ravens use the area quite frequently. Because I'm out there, I can't necessarily confirm it, but there are 5 ravens that, or at least I consistently see 5 ravens every time I'm out there, and every time I'm out in the wetland they're using. We have a couple of the big trees that they use, and then they also sit on the fence posts quite often. And then the admin, are you finding more diversity in mammals? So I am doing small mammal research. Currently, I've seen a pretty big decline in abundance for our small mammal populations. Part of the reason I've seen less small mammals is because the traps that we use are called Sherman traps, and we put bait in the back of this kind of box style trap. The small mammal goes in, the door closes. Unfortunately, it's only about this big, so a raccoon arm can fit into the small mammal trap, and they can take the food out and either trigger the trap or just take the bait away. So that's something, as of now, the data says that there's less abundance. There is more diversity, but then I have to account for those raccoons that are adding to bait going missing.
All right. If there aren't any other questions, then we can transition to our next graduate student speaker.
Thank you, everybody.
Yep, thanks.
Thanks, Andrew.
Good luck, Spencer.
Can you hear and see me, Hannah? Someone?
Yep,
Good?
you're good.
All right, great. Thanks for having me back. As Andrew did, I also talked at this meeting last year. So this is kind of an update, but my name is Spencer Parkinson. I'm a grad student at West Virginia University, as well as a part-time Forest Service employee in the Forest Health Program here in Morgantown, West Virginia. And I'm going to talk to you all about exploring birds as a possible vector for a novel forest pathogen, beech leaf disease. And this is normally where I'd ask people if you've heard of beech leaf disease to raise your hand. That's hard to do over Zoom. But if you have, throw in the chat, say yes. Hopefully, if you tuned in last year, you've heard of it. But I'm still just going to give you a brief overview of what it is. So what is beech leaf disease? It was first observed in Lake County, Ohio in 2012. It primarily affects younger trees and saplings. And the early stages of beech leaf disease is this darkening between the veins of the leaves. That eventually turns into more of a chlorotic striping and a puckering of the leaves in the intermediate stages. Advanced stages is this increased curling and leathering of the leaves, which then typically, in most cases, leads to defoliation. And within 2 to 7 years defoliation typically results in, results in mortality of the tree. And that's been observed in both saplings as well as older trees. So what we're trying to figure out is what causes beech leaf disease. In 2020, a novel nematode, Littleanchus Kanata McCanny, which I will call LCM from now on, so I don't have to say that every time, was first described by Lynn Carta, who I will talk about throughout this presentation. And it has been observed to be highly associated with beech leaf disease. So in a 2020 study, they took leaf samples from symptomatic beech leaf trees, or beech leaf disease trees, and in 45 to 90% of those leaves they sampled, they found this LCM nematode. They did the same experiment with the buds of the trees, and in over 90% of the buds, they also found this nematode LCM. So it's thought to be a causal agent. There's thought to be a few other things. There's a bacteria highly associated with called Wollenbachia, but that's beyond all of this. Here is a current map of the range of beech on the right, our American beech. And then on the left is the map of where beech leaf disease has been detected on a countywide basis. So we're going to zoom into that real quick. This is as of February 2023. And as I said, it was first discovered here in Lake County, Ohio, right on Lake Erie. And that's like the lighter beige color. Moving to the blues, which is 2015, 2016, 2017, to the dark purple. And the bright pink is 2022. And so as we can see, it's definitely been discovered or detected in much higher numbers recently. And this map isn't meant to be as an interpretation of the spread. This is more of a interpretation of the detection of beech leaf disease. But the interesting thing is it does appear to be in these isolated patches, as there's one county in Ohio, there's 2 counties in Virginia. It's up here in Maine, not connected to anything. And so we're trying to figure out, and also right here, it seems to not go south of the Mason-Dixon line in Pennsylvania for some reason. It doesn't want to go into Maryland. And I gave a talk in Maryland a few weeks ago, and I originally had thought that maybe the reason they weren't detecting it was because of lack of effort of detection, but they have done hundreds and hundreds of surveys to look for beech leaf disease, and it just doesn't seem to want to go south of there. So that's something very interesting. And that leads us to the question is, how is it spreading? And this is why everyone's here. We're thinking birds. The 2 main methods of dispersal that we are exploring through birds is ectozookery, which is just a fancy word for the external attachment to the feet, feathers, beak, bill of any bird, be it just the nematode itself or a mite that might contain the nematode. And then the other one is endozookery, which is just another fancy word for consumption, digestion, and excretion of successful propagules or nematodes or eggs of nematodes. And that picture at the bottom right is a tufted, tufted titmouse with a beech bud in its beak. And so as we know, birds are dispersals of many things. We know they disperse seeds. They can successfully disperse eggs of invasive carp species that's been observed in Europe. They can even ingest and disperse invasive brine shrimp. And another very common thing that, especially in the United States here, and dealing with nematodes, is the potato and soybean cyst nematode, which was a major agricultural problem. But the nematode that we're dealing with is not a cyst nematode, so it doesn't create like a callus layer on the outside of the eggs. So a question that we wanted to answer is, can the nematode we're dealing with survive the pH of a bird's gut? And so Lynn Carta, who I talked about earlier, ran some pH tests where she exposed the LCM nematode to different levels of pH for different amounts of time. And at a pH of 2, which is very, very, very acidic, I believe 70% of the nematodes, or 75% of the nematodes survived. And this was after being exposed for 24 hours. And then even at 1.7%, or 1.7 pH, sorry, which is more acidic than you're going to find in any sort of vulture's gut system, or the lower end of a vulture's gut. 10% still survived after 24 hours. So we figured the nematodes that we're dealing with are not going to be, they're not going to be sitting in a gut for 24 hours. So we went ahead and figured out, or went inside, that most likely they can survive that acidic environment. So I wanted to explore, can we detect the LCM DNA in the feces of wild birds in beech leaf disease areas? My second objective is, can we detect LCM nematode DNA in the feces of caged birds that we feed infected beech buds? And as I mentioned earlier, the beech buds that were infected had a 90% chance of having the LCM nematode. And then lastly, for ectozookery, or the external thing, is, can we detect LCM externally on wild birds in beech leaf disease areas? And so my two study sites are the Allegheny National Forest up in Pennsylvania. And as we saw on the map, pretty much all of Pennsylvania has beech leaf disease at this point. And then also the Holden Arboretum in northeastern Ohio, which was essentially ground zero for the spread of beech leaf, or for the detection of beech leaf disease. It's right next to Lake County, Ohio. And these sites were selected because they both have extremely high levels of beech leaf disease. And I did my sampling in the winter of 2021 and 2022, and a little bit of sampling in the summer of 2022, but the rest was in the winter of 2022 and 2023. And the reason we did it during the winter is because beech trees keep their buds throughout the entire winter. And so our thought is, as birds run out of other resources they might utilize, they might start to utilize beech buds as a food source. So my field methods were, we established bird feeders in places that were heavily infected with beech leaf disease. We set up game cameras at these bird feeders to, one, see if birds were coming. That's important. To see what kinds of birds we were getting at these feeders. The next step was mist netting. I'm sure most of y'all are familiar with mist netting. We set up mist nets around these bird feeders to catch birds. And there's a few birds we caught. We caught a brown creeper, hooded warbler, white-breasted nuthatch, amongst many others. From there, we did some sample collection, and I'll go into that in the next slide. We did a few different methods of sample collection. We analyzed the samples in a lab, and then we ran PCR, which is just a method of checking to see if we can find the DNA of certain animals. And the DNA we were looking for was that of the LCM nematode. So if we found positive samples of that, then we figured that it was present in the fecal samples. And so for my sample collection, we used three methods. The first one was the paper bag method, which is very simple. You catch a bird in the mist net, you put it in a brown paper lunch bag, and typically that scares the shit out of it. And you release the bird, you collect the fecal samples from the bag, and then we process it through the DNA, through the PCR. And that's testing for endozookery, or the internal process. Another one is diatomaceous earth. This is an external pesticide. It's used in a lot of poultry and other, I think, like for poultry mostly, but it's a mechanical pesticide that essentially slices on a microscopic level ectoparasites such as mites and nematodes to desiccate them. They'll die and they'll fall off the bird, and we can collect that on our sampling surface and then run a PCR on that. And then the last one, which we've just most recently started doing, is catching these birds, keeping them in cages for no more than 6 hours. And then we feed them infected beech buds from trees we know have beech leaf disease. And then we collect all the fecal samples from that, and we run the PCR. So here's just a quick rundown of what my results are so far as of, I think it's December. We've caught and taken samples from 139 birds. We've caught way more than that, but this is how many we've actually taken samples from, be it external or internal fecal samples or feather samples. 21 different species, well over 200 samples. and we've had, I think at this point, around 15 positive detections of the LCM nematode in our samples. So the graph just gives you a quick rundown, and it's what you'd kind of expect from feeder birds, because we are using bird feeders. We have a lot of chickadees, juncos, titmouse, goldfinch, nuthatches, sparrows. And here's just kind of a rundown of how our detections have worked monthly. The green is our negatives and the orange is our positives detections for the DNA of the LCM nematode. And as you can see, October 2021, that's when we first were establishing. We caught 2 birds that whole month, no positives. January 22nd, 2022, we caught 51 birds, no positives. March 2022, we caught 6 birds with 1 positive. That was exciting. And then April 2022, 10 birds, 3 positive, 40 birds. In August, 1 positive. November, we had 18 birds with 5 positives. So I'm not sure if this is an example or if this is showing seasonality or just us getting better at doing the experiments and figuring out how everything works, because this is all very new. But there's that graph. And most recently, we started doing these cage experiments where we keep them in cages and we feed them, and it's a much more controlled method of trying to see if it's possible for the nematodes to survive the digestive system. And what we did find is after keeping birds for less than 6 hours and feeding them only nematodes or only beach buds that had the nematodes in them, we were able under a microscope to observe the nematode, observe eggs of the nematode, and we were also able to observe that birds were actually consuming the buds, even though it was only the only food source they had. But as you can see in that picture to the left, you'll have to believe me, it's not a great picture, but that is the nematode that is highly associated with beach leaf disease, the LCM nematode. And we were able to observe that just straight from the fecal sample of, I believe, that titmouse that we had on the top right there. And so why am I doing this? What are we trying to figure out? We're trying to figure out how a nematode is spread over such a long distance and is so disconnected. Nematodes are aquatic animals. They need a film of water to travel, and they don't live. I mean, there's many generations in a single year. So we want to figure out how they're traveling all these long distances, how they're going to all these different places that are seemingly disconnected. And of course, we always think humans are the vector, excuse me, but the places that we're typically finding first discoveries of beach leaf disease are very rural and not near urban areas and not typically visited by many people. So from there, if we do discover that birds are a vector for it, we could figure out which birds specifically are spreading it, and then maybe be able to make predictive models as to where the spread will continue to happen based on migration patterns and movements of different species of birds. And I hope most people at this point have heard of beach bark disease. It has already decimated our beach populations, and beech is a very important tree species for animals. It feeds bears, grouse, white-tailed deer. It's a huge, important hard-mass species. And trying to figure out how we can maybe help it along a little more is very good. And finally, just moving forward, the next steps are viability. And I said we had observed the nematode in the feces, and we've had positive DNA tests for the LCM nematode, which is associated with beach leaf disease, but we're not sure if it's alive. So under microscopy or under the microscope, we see the nematode, but it's not moving. And when we do the PCR, we are not sure if it's a dead nematode that we're getting DNA from. So we haven't confirmed that viable nematodes are successfully passing through or externally being transferred on birds and are able to reproduce and spread the disease. So that is the next step we really want to explore. Environmental factors is maybe just seeing how different climate changes in areas or temperature changes in areas might be affecting the levels of beach leaf disease. Something that no one's looked at yet is the nematode load of beech nuts. And as I said, they're a very important food source for animals, so it's hard to even find beech nuts to test for the nematode. But that would be another one, and that would open up a whole new can of worms as the other vectors, such as bears and white-tailed deer and grouse. And then the final thing we're looking at, it's a new study coming out of Penn State in the next couple of years, is caterpillars. They have found the LCM nematode in caterpillar poop or caterpillar frass. And so exploring if those caterpillars are feeding on infected beech leaves and then possibly being consumed by a bird, the caterpillar might act as a buffer to protect the nematode as it passes through the gut of the bird and potentially being spread elsewhere. So yeah, that's all the exciting things we have coming up. That's all I have for you. I'd like to thank the Forest Service, West Virginia University, Holden, and Forest Gardens Arboretum. And I will take any questions.
Great, thanks, Spencer. So yeah, throw your questions in the chat if you have them. If you want to ask your question through video, feel free to request that or raise your hand and we can unmute you. But it looks like there's a couple of questions in the chat.
What are they? Okay. Was that Megan? Have you extracted DNA and confirmed the nematode using PCR and genetics methods from the fecal samples from nematode infested bud-fed birds? Yes, we have. Short answer, but yes, we have confirmed it from caged birds that we've fed buds to, and we have sequenced the DNA to prove that it is the nematode we're looking for. Is there a way to help infected trees? Yes, it's extremely expensive. There's nematicides that we're looking into. There's a lot of research being done, and it's probably typically going to be only applied to high value ornamental trees in cities and like arboretums. But there is nematicides. They're just very labor intensive and expensive. Limited to feeder birds at the moment. Yes, definitely. The question is there a reason to think that nematode might be spread by other species beyond what you can capture 100%? As far as I know, we haven't observed birds in the wild consuming beech buds, but we think they are, and also just the interaction between beech leaves and birds might be spreading the nematode or mites containing the nematode to the feathers. And as I mentioned with the beech seeds or beech nuts, that might be being consumed by grouse or other species of bird, I think blue jays as well. So yes, definitely. Any pattern in species for birds that tested positive for nematodes? So far we have downy woodpecker, American goldfinch, and the most predominant one, as you can see here in my question slide, is tufted titmouse. So those have been the ones that we found the majority of them. Yes, it pretty much is. It is pretty much, the question is it lethal to all trees that are infected? Yes, eventually. Doesn't matter really the age. As I said, they do affect younger saplings. But once the defoliation starts occurring, it's typically 2 to 7 years until this complete defoliation occurs.
All right, well, if that's all the questions for Spencer, then we can I guess move on to me. Thank you, Spencer.
Thanks, everyone.
All right. Okay, so just if someone can give me a quick heads up if you can hear me and see my slides.
Yes, Hannah, we can hear you and your slides look great.
Okay, all right. So I'll go ahead and get started. So just to briefly introduce myself to anyone who might not know me already, my name is Hannah. I'm a board member of the Mountaineer Audubon. I was a past president, and I am a PhD candidate in my 5th year pursuing a degree in natural resource science at West Virginia University. I'm working with 2 co-advisors, Dr. Peter Wood, who was a federal scientist in the West Virginia Cooperative Fish and Wildlife Research Unit and is now retired, and Dr. Chris Rota, a professor in the WVU Division of Forestry and Natural Resources. So for my dissertation research, I'm focusing on the effects of climate change and landscape change on forest bird communities, abundance, and nest success in the Appalachian Mountains region. So before going into the details of my research questions, I wanted to start off with a kind of broad overview of why I'm interested in this topic. So many of you may have heard of the study out of the Cornell Lab of Ornithology back in 2019 that found that we've lost about 2.9 billion birds, which is nearly 30% of the total bird population in the USA and Canada in just the past 50 years. And of those, 170 million were eastern forest birds, including one of my species of interest, the wood thrush. One factor that is thought to be contributing to declining populations of forest songbirds is global climate change. So we've seen temperatures in the USA get warmer within the past 50 years, especially in the Northeast, which is also where many bird species are thought to be at risk due to climate change. So you'll notice that the Appalachian Mountains in particular harbor a high number of climate-threatened bird species, according to the National Audubon Society. And focusing on the Appalachian Mountains, they serve as the southernmost limit of many species breeding ranges in the eastern USA, such as black-throated green warbler, which is pictured here with their range map, as well as other birds that usually breed in the boreal forests of the Northeast and Canada, but then have these trailing edge populations within the higher elevations of the Appalachian Mountains. However, this region is also experiencing rapid land cover change in some areas, and it's important to see how landscape change interacts with the climate concerns for birds in the Appalachian Mountains. So the purpose of my research is to better understand how the changing environmental conditions in the Appalachian Mountains in turn influence forest songbird communities and populations. To look at long-term trends over time, I have access to several historical data sets, including nest monitoring and point count survey data from the Monongahela National Forest and nearby private lands within the Central Appalachians, and point count survey data from collaborators from the Northern and Southern Appalachians. And then from 2019 to 2021, I also contributed to new survey efforts within the Central Appalachians. So collectively, these historical and new data sets are being used in the 4 chapters of my dissertation. So for my first chapter, I looked at the relative influence of climate versus land cover change on 14 forest songbird distributions in the Appalachian Mountains, and then projected those distributions based on future climate and land cover scenarios. I was particularly interested in figuring out whether climate or land cover change has greater effects on bird distributions in this region. So here I have a graphical abstract of my results for black-throated blue warbler, but I will be talking more about my overall results in just a few minutes. So we're just gonna keep moving on. For chapter 2, I'm using long-term historical point count survey data from the Northern, Central, and Southern Appalachian Mountains to understand why forest bird communities are changing over time and their relationships with temperature and precipitation across elevational and latitudinal gradients. So this map on the right shows where my 3 study regions are located within the Appalachian Mountains, so Northern, Central, and Southern. And the Appalachian Mountains is indicated by this shaded gray region. And then these plots at the bottom show some of the results for trends in wood thrush abundance over time. So here we see that wood thrush abundance has been decreasing in the northern and central study regions, but not so much in the south. And then the rate of decline is steepest at lowest elevations. For chapter 3, I shift focus from climate factors to concentrate on the effects of forest management. So I'm examining long-term responses in bird communities, abundance, and nest success in the Monongahela National Forest, which is predominantly unharvested landscape, and in a nearby industrial private forest, which represents a landscape that has a history of intensive harvest. And then finally, for chapter 4, I aim to understand which bird species are using wildlife openings within the Monongahela National Forest. So wildlife openings are areas of early successional habitat, so including grassy or shrubby areas that are surrounded by mature forest. And my goal for this is to identify the characteristics of wildlife openings that support a diversity of game birds, such as wild turkey, ruffed grouse, and American woodcock, as well as breeding and post-breeding songbirds, with the ultimate goal of improving and guiding management of the more than 2,000 wildlife openings within the Mon National Forest. And I also wanted to briefly mention a side project that was somewhat related to my dissertation research. So last year, I published a paper on bird communities and individual species that are strongly associated with red spruce forests in West Virginia. And so here's a graphical abstract of those findings. So we found that 11 bird species were consistently and strongly associated with red spruce. And then 3 of those, the magnolia warbler, dark-eyed junco, and blue-headed vireo, were particularly associated with 10 to 50% red spruce cover. And then the other 8 were particularly associated with more than 50% red spruce cover. And then 3 of those, the yellow-rumped warbler, golden crowned kinglet, and black burning warbler, were often found in stands with older, larger trees. So in the past, so I've given this I've done the Science in the Scope program for, I think, the past 3-4 years. So I've talked about bits and pieces of my 4 chapters before and that side project. But for this rendition, I decided to give a modified version of a presentation that I gave during a Wildlife Society conference. And so this is covering the full results from my first dissertation chapter. And that's the one that focuses on potential effects of climate and land cover change on forest songbirds in the Appalachian Mountains. So as I mentioned at the very beginning of this talk, climate change may play an important role in influencing bird species. Warming temperatures and altered precipitation trends are predicted to have long-term effects on bird distributions and abundance in the future. For example, these maps from the U.S. Forest Service Climate Change Bird Atlas show the modeled distributions of black burning warblers in the eastern United States currently and in the future under a high emissions climate scenario. And you can see that the overall range for black burning warblers is predicted to shrink, and regional incidence is predicted to decrease. In addition to temperature and precipitation, recent studies of bird responses to global climate change are increasingly incorporating land cover change as an additional factor, since we know that land cover change that results in habitat loss and conversion can have negative effects on birds. The consequences of both climate and land cover change on bird communities in mountainous regions are of particular conservation concern. So there have been studies from mountain ranges across the world focusing on montane and high elevation bird species, which are thought to be at the greatest risk from global climate change. And building off of these previous studies, I wanted to know, how might a changing climate and landscape influence bird distributions in the Appalachian Mountains, a temperate mountainous region in the eastern United States that functions as the southernmost limit of several bird species ranges? High elevation areas with cooler climates in the Appalachian Mountains allow for trailing edge populations of species that otherwise tend to breed at higher latitudes, such as black-throated blue warblers, least flycatchers, and veeries. All 3 species primarily breed in the boreal forests up north, but their breeding ranges do extend down the Appalachian Mountains range. Because they tend to be restricted to higher elevations in the Appalachian Mountains, they may be particularly sensitive to a changing climate in this region. And we know that the Appalachian Mountains are expected to become warmer and wetter in the future. So these are maps of contemporary and projected future temperatures and precipitation for the month of June. So that's the prime time for birds to breed in much of the Appalachian Mountains bird conservation region, which is the area outlined in black. Land cover is also projected to change within this region, again outlined in black, with declines in forest and agricultural land cover and increases in urban cover over time. Therefore, the purpose of this study was to combine climate and land cover metrics with bird survey data to quantify the influence of climate versus land cover on the relative abundance and distributions of 14 forest songbird species in the Appalachian Mountains, and then project future distributions in 2100 based on expected scenarios of climate change and land cover change. So our study area was the Appalachian Mountains Bird Conservation Region, which is shaded in dark gray in this map of the United States. And I focused on 14 forest songbird species that use mature forest as primary breeding habitat. And I assigned them to one of 3 climate classifications. So 5 species were cold-associated species, including the black-throated blue warbler. So these are species found at primarily at higher latitudes and higher elevations within the study region. 4 were warm-associated species, including summer tanager, primarily found at lower latitudes and lower elevations within the study region. And then 5 were just climate generalist species, including the red-eyed vireo. So they're found just throughout the study region. So I obtained 20 years of count data for the 14 individual bird species from the North American Breeding Bird Survey using 322 routes found within the Appalachian Mountains Bird Conservation Region. The data consisted of the bird species and numbers detected during 3-minute point count surveys from 50 stops located along each route. For each BBS route, I calculated the total amounts of each of the 14 focal species in each year to obtain 20 years of route level annual total species counts. And that was the main focus for this study. For the contemporary climate data, I compiled monthly temperature and precipitation data corresponding to each year and route. And I had 4 climate variables total: mean temperature and mean total precipitation during March and April, which is the vegetation growing season, and May and June, which is the bird breeding season. And then my 2 temperature variables did end up being correlated. So I replaced the mean growing season temperature with the difference in temperature between the growing and breeding seasons. And then for contemporary land cover, I calculated the proportions of 3 land cover categories: deciduous and mixed forest, conifer forest, and developed land. And then to project into the future, I created a hexagonal grid that encompassed the study region. And within each grid cell, I summarized the contemporary and future environmental data. So looking at future conditions to calculate the mean climate conditions in 2100, I used long-term monthly data from 3 climate models and 2 greenhouse gas emission scenarios. So I averaged the 3 climate models to yield an average low emissions, ALE scenario, and average high emissions, AHE scenario. And then in addition to the 2 averages, I modeled the coolest scenario and the warmest scenario, and those represent the 2 extreme possible outcomes from the climate analysis. And then for future land cover, to calculate the future proportions, I used USGS projections, which also incorporated a low and high greenhouse gas emission scenario. And then to model all the data, I modeled each of the species individually and explored how the response variable, which was the route level annual total counts from the breeding bird survey, how those were affected by the predictor variables, which included latitude, elevation, the 2 temperature variables, the 2 precipitation variables, and then the 3 land cover variables. And so to project the future distributions, I used the model to predict contemporary and future species counts to the grid that I created covering the study area, using data from 2000 and then using data from the 4 future climate and land cover combination scenarios. So the cool, the ALE, AHE, and warm. And this is an example for black-throated blue warbler of doing that. And then I quantified 3 indices of changes. So differences in the total projected count of individuals across the entire study region, total number of occupied grid cells across the entire study region, and then shifts measured by distance and angle in spatial distributions between 2000 and 2100 for each of the species. So getting into results, land cover variables tended to be more often significant for the 14 focal species. And in addition, the most frequently important predictor variable was proportion of deciduous and mixed forest, which was significant for 10 of the 14 species. Changes in land cover, especially concerning proportions of deciduous and mixed forests and developed land, seemed to more strongly influence relative abundance than either temperature or precipitation. And that can be seen in the results for scarlet tanager, which is one of the 5 climate generalist species. And here I have the full results for black-throated blue warbler, which was one of the 5 cold-associated species. So here you can see none of the climate variables were significant, but proportion of deciduous and mixed forest had a significant positive relationship, and proportion of developed land had a significant negative relationship. In terms of projected changes in relative abundance from 2000 to 2100, here I have color-coded the direction and magnitude of percent change, such that red indicates a decline, gray is little or no change, and blue indicates an increase, with bigger effects indicated by darker shades. So the first set of 5 species are the cold-associated species, and you can see that the results are a bit variable, but 4 of the 5 are predicted to experience declines of at least 2% in at least one future scenario. Of the warm-associated species, which are the 4 grouped in the middle, and then the climate generalist species, which are the bottom 5, the vast majority show very little predicted change in relative abundance. So here's results for black-throated blue warbler again. You might notice that the highest increases in relative abundance, which are indicated by the teal colors, are concentrated in the northern half of their range. And then the steepest decreases, which are indicated by the orange colors, are concentrated in the southern half of their range. And this was a really interesting regional pattern that actually held true for 3 of the 5 cold-associated species. Looking at overall range, there really weren't any statistically significant differences for any of the species. And this was looking at the total number of occupied grid cells across the entire study region between the 2 time periods. So most species really didn't show much percent change at all. And the one that showed the most change was black-throated blue warbler, which shown here, you can see the modest range gains in 2100. And mostly in the northern part of their range. And then there were also no statistically significant shifts in the spatial center of the projected species distributions from 2000 to 2100, but there was an interesting trend among the climate classifications. So in this table, I've color-coded the distances of the shifts, with the darkness of the blue color indicating greater distances. And you can see from the color patterns that the greatest shifts in distance were experienced by the cold-associated species, followed by more modest shifts by warm-associated species, and then very little change for the climate generalist species. And then in this table, for the shifts that exceeded 50 kilometers in distance, I color-coded the direction of the shift. So towards the northeast or north is blue, towards the east is purple, and towards the south or southwest is red. And with the exception of Viri, most of the shifts are in the northeastern or eastern directions. And then the maps for the shifts in the spatial centers of projected species distributions weren't super impressive to see. Most of the time, the shifts were pretty small. But here's the example of black-throated blue warbler, where the dot is the spatial center in 2000, and then the yellow represents the spatial center in 2100. And you can see that northeast shift. So to summarize everything, proportions of land cover types tended to be more influential and had higher effect size than temperature or precipitation amounts. Cold associated species were projected to experience slight declines in relative abundance, with the steepest declines concentrated in the southern portion of their ranges within the Appalachian Mountains Bird Conservation Region. But there was little projected change in their overall ranges. In addition, the spatial center of projected distributions for cold associated species did shift the greatest average distance with consistent movement in the northeast direction. Meanwhile, the relative abundance and ranges for warm associated species either declined or remained similar in the future, with modest distribution shifts in either the northeastern or eastern direction. And finally, climate generalist species appeared to be least affected by future climate and land cover changes. There is little to no change in overall projected relative abundance or range, and very little to no shifts in their distribution. So at a broad spatial and time scale, the net projected impact from climate change in this region was pretty modest. And perhaps that's because the mature forests and elevational gradient of the Appalachian Mountains might be able to buffer the impacts of climate warming for forest songbird species. Other studies have found that dense forest canopies result in cooler microclimates, and cooler temperatures can be found at higher elevations. So thinking about conservation and management implications of these findings, it seems like potentially good news that the projected impacts of breeding season climate change on our focal forest songbird species were minimal at a very broad scale in the Appalachian Mountains, but land use changes may pose a more immediate threat to birds in this region. So before I end, I would like to acknowledge and thank my funding and support, as well as the sources for the data that I used. And then I will end here. I'm happy to answer questions. And if you want to discuss anything about my other research projects, please feel free to reach out. Okay, and like with the other speakers, feel free to put questions in the chat. And I guess if there are no questions, oh, wait, okay. Okay, so would you mind just showing us the list of which species are warm versus cool versus generalist? Yeah, so let's see, let me go back to that slide. Okay, hold on one second. Oh, there. Okay, so let's see. So for cold associated, I had black-throated blue warbler. Let's see. I'm trying to remember off the top of my head. I do have a full list somewhere. Okay, so yeah, so black-throated blue warbler, least flycatcher, veery, I think Blackburnian warbler, and then there was one more. Which is not coming to mind right now. Warm associated species, I had summer tanager, worm-eating warbler, Swainson's warbler, and one other species. Man, I'm forgetting one for each one. But then climate generalists, red-eyed vireo, wood thrush, scarlet tanager, and then two others. I can send you, if you reach out to me, I can send you the full list. But yeah, so I had those 3 climate classifications and then total of 14 species. All right, well, if there aren't any other questions, Jane, you can feel free to take back over.
Well, Hannah, that was really great. What a massive amount of data you worked on. Oh my gosh, made my head swim and I didn't even have to do any of it. I did want to say earlier there was in the chat that the Woodcock Walk this coming Saturday is probably already full. So make sure you look into that before you plan to attend. And thank you to all the graduate students. It's really interesting what you're doing. And let's think of great ways to get all this information out to the rest of the world. So they're on the same page as us. And one more spring talk in the month of April. And then we'll take a hiatus for summer and start back up in September. So thank you everybody for attending this program. Especially thank you to our speakers. Oh.
Thanks for having us.
Oh, you're welcome.